Good morning, good afternoon, and good evening, everyone. Welcome to this WuXi AppTec online lunch event, Propelling Boundaries, Innovative Technologies in Early Drug Discovery. I'm Jingsi from WuXi AppTec, and I'm honored to be the host for this event. Today, we're excited to share with you many of our latest advancements in the field of early drug discovery, including the new DEL and HTS solutions, direct-to-biology applications, new modalities like oligo and peptide discovery tools, and comprehensive target discovery capabilities. Joining us today are three experts from WuXi AppTec. We have Dr. Wenji Su, the head of Early Discovery Platform, will update with you the small molecule drug discovery platform. Dr. Andy Ng will bring you the integrated oligonucleotide drug discovery platform, and Dr. Zhongyao Ma will present the innovative and the latest technology updates in the peptide drug discovery and also the target discovery. So next, please enjoy the very enriching talk. First, let's welcome Wenji.
Hi everyone. Today I'll be talking about the overview of the upgrades of our platform, as well as focusing on the small molecule part of my of the platform. So first of all, I introduce like the evolution of the food finding platform in WuXi. So we started expanding our capability in this space since 2017, and in 2017 we built our first single automation system for HTS, and we built our first cell, which is about 0.1 million. And it's been a remarkable journey since then. Over the last 8 years, we evolved from only having such, the first library, um, from the classic small molecules to different modalities. And until now, we have collected enormous libraries with a diversity over 1.1 trillion molecules. And we have also built a fully integrated automation system to support our HTS platform. And here's an overview of what the WuXi Biology hit finding platform looks like. As you can see that we now have built a very comprehensive platform covering all modality selections, from the smallest fragment to small molecule to covalent molecule to peptides, oligos, aptamers, and even antibodies. And for all those modalities, we have built the complementary screening platforms to help finding those hits from those libraries, from the more traditional HTS and DNA-encoded libraries to some of the newer additions like OBAC-DEL, mRNA display, and SELEX. And for each of those screening platforms, we aim to build the best in the industry to ensure that we can support our clients' project and we can deliver the best results possible in those hit finding programs. So today I'll be focusing on the upgrade of our small molecule drug discovery platform, while my colleagues Andy and Zhongyao will be focusing on oligos and peptides and other modalities. So for the small molecule platform upgrades, I'm gonna focus on three aspects. One is the DEL platform, the second is the HTS platform, and the third is the direct-to-biology platform. And for those platforms, we will introduce the new generation of our libraries and screening methodologies and our effective ACR solutions. So first of all, the DNA-encoded library platform. So here is a record of we have achieved so far for this platform. So for the DEL Pro product alone, we have been screened over 600 different client targets, ranging across over 10 different popular target classes. And the most advanced hits that we know has been into the phase 3 clinic trial. And the most strongest hits, the primary hits is about 300 picomolar in affinity, which is quite remarkable considering it's just a primary hit. And we have also accumulated over 40 publications over the years, spreading through like discovery of new ligands, new chemistry, screening method, informatics, and review and book chapters. This is just to show the devotion of the new technology development into this platform. So for today's upgrade, I'll be focusing on three directions: the novel chemistry, new modality, and new screening solutions. So for the new chemistry, we have we have been keep doing developing the chemistry, the libraries, And here are some highlights of the newer achievement we have incorporating our new products in Dell Pro and Dell Lite. So for the Dell Pro libraries, we have currently um, reached about 40 billion chemical diversities across around 200 libraries. And among those libraries, we have been put a strong emphasis on adding more 3 and 2 cycle libraries. So currently we have over 90% of our library collections 2 or 3 cycles, with only minimal percentage of 4 cycle libraries. And about over 20% of those newly added building blocks are brand new in the collection that has never been put into library. And we aimed and we managed to incorporate our proprietary institute information reaction into the libraries as well. And the new features for the Dell Lite libraries is that we have reached 10 billion compound diversities for this library collection and reached about 70 different libraries. And out of that 70 libraries, 32 of them are completely new, adding in about 8 billion new compounds, which is like 80% of the entire collection. And we also incorporated the new heterocyclic libraries into the collection. And for this, we also reached about 90% of these libraries are 2 cycles or 3 cycles. So I guess the key message here is that the upgrade is significant, and we aim to bring the new almost like completely new products into the collection, and let our client to be able to identify novel chemical matters out of those libraries. And one thing I want to highlight is that for the new 3 cycle libraries, we had a strong emphasis on focusing a lot on drug likeness. So with our, you can see that with our 5 compliant molecules, we increased by 21% to 32%. And the molecular weight property and the stereochemistry property has greatly improved over the last generation. So you may ask, how do we reach such new chemistries? And what we did is that we routinely explore new hot molecules in the market, like new cores, new pharmacophores, new building blocks, and we develop new reactions, try to bring those new building blocks into our libraries. And apart from publishing those new reactions into peer-reviewed journals, we also aim to bring those build those molecules and chemistries into our new library collections, such as the Dell Pro collection. Here's an example that we developed in situ reinformation reactions. Not only we got a publication out of it in the JOC, we also added it to the new collection of our screening service product. So apart from new chemistries, we also put a lot of effort into the new modality development in terms of the library collection. So here are some highlights of new modality DAOs supporting innovative therapeutics. So for COVID, we have highlighted the covalent DAOs, peptide DAOs, molecular glue DAOs, PROTAC DAOs, and RNA targeting DAO. For the covalent DAO, we have built in over 100 different electrophiles with balanced activities targeting not only cysteine, but lysine, tyrosine, and serine. And the entire collection is now 1.6 billion. And for the peptide DAO, we incorporated over 500 natural amino acids with specially designed backbones. And the library collection is now 300 billions. And for the molecular glue DAOs, we emphasize on the cereblon binding codes with validated sites and modifications. And apart from that, we also built collections on VHL and other E3 ligands like HIT4. And the current collection of molecular glue DAO is about 5 million. And for the PROTAC DAO, we allow simultaneous exploration and optimization of linkers and POI binders, and the collection is now 4 billion. And for the RNA targeting DAO, we link compounds featuring RNA binders and utilizing our brand new DAO zipper technology, which I'll talk about in the later pages. And the current collection is 100 million. And apart from the new chemistry and new modalities, we also keep on developing new screening solutions to allow for more efficient and more accurate delivery of the screening results. So for the new modality DAOs, we develop complementary new methodologies for the screening to help effective identify hits from the new library connections. For example, for molecular glue, and we optimize our dual pull-down method to allow for simultaneous detection of ternary complex formation. And for the covalent screen, we optimize our incubation time and elution methods for a single round of selection. And then this allows for better signal to noise ratio. So and also for allow assessment of different time points to allow for different assessment of different axial file activities. And for Del Zipper, which is a novel screening method we brought to the platform, it allows for minimizing non-specific binding between DNA barcode and the target. And this allows for expanding DNA-encoded library applications, which I'll talk about in the next page. And apart from auto screening method, we also developed on-DNA validation platform, allow for parallel synthesis of on-DNA molecules that mimics library synthesis routes and allows for recapitulating all the byproducts and partial products and for dissecting of the binding components during the hit conformation stage. So we want to have a special highlight on the Del Zipper and why we think it's a very important part of our platform because Del Zipper solves two essential problems in the Del technologies that has been in the past. One that the double the single-stranded Del that we currently most people are practicing um, allows for more efficient PCR and amplification. However, with the single-strand region, there could be secondary structures, and also allow for hybridization with different sequences, so that it does opens up more non-specific bindings between targets and the DNA barcode. However, with the double-stranded blunt end Del, the PCR efficiency is something that's a problem. So with the new Del zipper we brought into place, we not only converts the single-stranded region into a double-stranded region, so it reduces like non-specific binding, and then we also make it convertible to single during the PCR step, so that it allows for comparable PCR efficiency to partial single-strand Del barcode, and is significantly higher than the fully double-strand blunt and Del. And what's more important is that this technology is compatible with our existing Del library, and it allows for screening against conventional protein, RNA, DNA binding protein, surface, and anything that you can think of. And we think it's a universal solution for all Del practitioners, and we are welcome to have anyone who's interested in trying this technology in their targets to contact us for a trial. And apart from the Del, I'm also going to talk about our new upgrades to the high throughput screening platform. So we'll be focusing on three aspects: the library upgrades, the assay expansion, and automation. So first of all, the library. So we have now built a 370K small molecule libraries for the HTS platform that we offer. And this library we curated so that it has high diversity with an average Tanimoto similarity score at about 0.12, and it's significantly less than many of the commercial libraries we can find in the market. And it also makes sure that we have still enough coverage and densities of the chemical space. And we also focus a lot on drug likeness with RI compliance molecule over 95%. And we also pay special attention to stereochemicals complexity. So we want to have all the different shapes of molecules as much as we could. And now we have over 30% of our compounds featuring a high FSP3 score over 0.4. And for the assay part, we expanded on the variety or the availability of assays in this platform. So we not only can do regular plate-based, biochemical and cellular screens, we now incorporated high content screening assays with target-based and phenotypic screens and allow for imaging-based readout. And we also incorporated the ASMS, is a affinity selection mass spectrometry, which allows for pulling up the molecules to really enhances the throughput and shorten the cost and the time, and it allows for minimal assay development so that with the complementary HCS and ASMS methods, we allow that our customer to find the best approach of the assay for their programs and allow for a more comprehensive solution tailored to the specific targets and modalities. And last but not least, we fully integrated our automation system into the HTS platform. And now we have built two sets of automation system. It allows for screening up to 400K data points per week. And we were able to miniaturize our screen routinely into 1536 well plates. And that allows significant reduction of cost by 50 to 75% comparing to the traditional 3D4 well plates. And with the fully automated system in place, we ensure precise treatment of each individual plates and allows for much better consistencies and across different plates and within the same campaign, and allows for much more reliable data outputs for the screening. And the last platform we're gonna share with you is the direct to biology platform, which is a new emphasis this year, and we believe is a really good early discovery accelerators for our customer. So what is D2B? It is actually a very cost-effective kit expansion SAR solution, and it has a special application highlights into the TPD molecule discovery, including PROTACs and molecular glue, and peptide optimizations and XCC explorations. It has three different key advantages. One is rapid evolution of the molecules, and it allows for like one cycle of optimizations within 2 to 3 weeks, and it allows for data-rich compounds. For example, one single FTE can produce 300 compounds within 3 days, and that allows for a very remarkable cost and time saving. With its plate-based and small-scale synthesis, can reduce about 75% of the cost compared to regular ACR programs. So here's just the breakdown of the workflow of what the D2B looks like. A simple answer is that it's a rapid design, make, test, analyze DMTA cycle. So we can, in parallel, design the library and develop the assay. And while synthesizing the compound, we can do the QC in parallel with the assay, and then do the assay analysis right after. And the entire cycle can be completed within 2 to 3 weeks, which is a remarkable saving over the regular SAR program. So here's an example of what the D2B result looks like. We can so this, on the left-hand side is a pie chart showing 300. It's a pilot campaign. Actually, we made 300 different compounds in one cycle, and then we were able to achieve the majority of the compounds over 60% of purity, and with over 60% of the compound that we make, that's over 90% purity. And we tested those compounds with and compare it to a pure product. And as you can see there on the right-hand side, it shows pretty good consistencies in terms of activity and ranking in terms of the for the pure product compared to the purified products. So it does allow us to have good confidence in this technology to be able to provide meaningful data for the SAR analysis and for next step guidance. And with that, I would like to just summarize here and then transfer to my colleague Andy to introduce the oligo part. Thank you.
Thank you, Wenji, for bringing the updates on small molecule discovery. Now let's transition to new modalities and welcome Andy to introduce the integrated oligo drug discovery platform first.
Thank you for the introduction and welcome everyone to my talk today. Today, I'd like to share with you our integrated oligonucleotide drug discovery platform at WuXi Biology. Our team can support the development of various kinds of oligonucleotide drugs, including ASOs, siRNAs, microRNAs, aptamers, etc. We can also work with the conjugated version of the oligo for delivery, as well as the packaged version, such as using LNP for delivery. Our team has accumulated a lot of experience in terms of helping to develop oligonucleotide drugs. We have worked with over 250 clients, worked on over 250 targets in various disease areas, including CNS, autoimmune, and metabolic. We have completed close to 1,000 projects, and many of these have been for supporting IND. So the capabilities of our team really span the full oligonucleotide drug discovery process, starting from target validation through oligo sequence design, through lead selection and optimization, and finally into in vivo studies, including PK/PD and early toxicity assessment. We offer you a one-stop service plus flexible modules to step in. So there are really three key challenges in the development of oligonucleotide drugs, namely efficacy, toxicity, and delivery. So in terms of toxicity, one key aspect of toxicity is off targets. With respect to each of these challenges, we have developed several solutions to address these challenges. To address the efficacy challenge, we have developed an in silico design platform that is meant to help you efficiently discover effective and safe oligo sequences. So the platform factors in various factors such as efficacy prediction using our in-house machine learning platform. We also factor in off-target prediction at the design stage, as well as cross-species activity. We also offer a patent search service to assess for IP risk of the sequences. Here are some results for our oligo design platform. So we compared our performance with an open source tool for design, and we designed sequences against two different targets, target 1 and target 2. As you can see from the diagram, the proportion of sequences that are highly active, meaning the inhibition ratio is above 50%, is much higher for our platform as compared to the open source platform. Although the proportion of highly active sequences differ between targets, but in general, our platform outperforms the open source platform. Here is a specific case study we did for sequence design. Here we designed 200 sequences targeting C5, which is an autoimmune target. After design and screening, we found that over 50% of these sequences are highly active, meaning that they knock down the target by over 50% at 10 nanomolar. And these sequences also had an EC50 of under 0.1 nanomolar. So from just one round of design and screening, we can manage to find many potent hits, thus basically helping to address the efficacy challenge. Moving on to the second challenge, which is toxicity. We have developed a suite of tools to assess for toxicity of oligonucleotide drugs. As mentioned, off-targets is a key aspect of oligonucleotide toxicity. To address this challenge, we have developed various tools to evaluate off-targets, including in silico tools, RNA-seq-based tools, as well as RT-qPCR and sidechain reporter tools. So these tools are applicable at different stages of the drug discovery process. For example, at the design stage, we can apply in silico analysis for off-targets and pick sequences with fewer off-targets to proceed for screening. Once we get several potent hits from screening, we can do RNA-seq experiments to basically assess for off-targets in vitro. Our team has accumulated a lot of experience in terms of looking at off-targets in different types of cell models, including cell lines, primary human hepatocytes, as well as iPSC-derived cells. Once we get a list of potential off-targets, we can further validate those using RT-qPCR as well as the sidechain reporter system. Our team has accumulated over 7 years of experience in terms of evaluating off-targets. In 2025 alone, we have completed close to over 4,000 samples to assess for off-targets. And around 50% of these projects are supporting IND filing. So we have a lot of experience in terms of supporting regulatory filing. Moving on to the third challenge, which is delivery. As everyone knows, delivery to the liver is more or less a solved challenge with the use of GalNAc as a targeting ligand. However, extrahepatic delivery remains a key challenge today. At WuXi AppTec, we have developed a suite of tools to enable extrahepatic delivery. This includes conjugation-based methods as well as carrier-based methods for delivery. In terms of conjugation-based methods, beyond GalNAc, we can conjugate the oligonucleotide to a peptide, lipid, antibody, or aptamer to enable targeting to tissues beyond the liver. For carrier-based methods, we can use LNP or lipid nanoparticle, exosomes, or other nanoparticles for delivery. If we need to discover novel ligands, the early discovery team offers various types of high-throughput screening approaches, including the DNA-encoded library, phage display, mRNA display, and CLEX to enable the discovery of novel ligands. Once the delivery tools are in place, we can further evaluate them using a suite of tools. So this page shows various in vitro tools for evaluating delivery. This includes binding of the ligand to the cell receptor by SPR, internalization of the drug molecule into the cell, and we can use FACS or high content screening on the right. And on the bottom left, once the drug is in the cell, it needs to get released from the endosome. So we also offer various endosome release assays, such as the immunofluorescence colocalization assay to look at endosome escape. Moving to in vivo, we can also evaluate the biodistribution of the vehicles by IVIS or live imaging. So the data on the top shows, the distribution of LNP particles of different formulations in the mouse, and we can quantify the distribution to various tissues on the right, such as to the heart, the liver, spleen, lung, kidney, lymph node, and muscle. At the bottom, beyond looking at biodistribution, we can also assess for PK and PD. So the data here shows an antibody oligo conjugate that is supposed to target the muscle. So you can see from the data on the bottom left that we can knock down the target gene in quadriceps muscle. And on the bottom right, we show distribution of the oligo conjugate in muscle. So that is basically a summary of our oligo platform as well as key capabilities. Thank you.
Thank you, Andy. So besides the oligo, another new modality, the peptides, also have gained significant attention in recent years. So let's follow Zhongyao to dive into the exciting world of peptide discovery.
Thanks, Jinsu, for the introductions, and it's my honor to be here to present our peptide drug discovery capabilities in this launch event. Today, I'm glad to share some of our recent progress regarding peptide DNA-encoded libraries, mRNA displays, and we also would like to share some of our recent R&D progress, which combine phage display together with peptide DNA-encoded libraries to further improve our primary peptide hit binding affinities. So first of all, our integrated peptide discovery platform has a lot of experience. So far, we have been finished more than 100 screening campaigns in which we have already successfully screened more than 70 different targets. And within these different targets, the best hits we have been identified is around 2-digit picomolar binding affinities. So why we care about peptide and why peptide is potential new modality compound in drug discoveries. As we can see in this slide, peptide lies right in the middle between small moleculars and the therapeutical protein and the biologics, which give it unique advantages such as highly potent and selective, less toxic, and also low manufacture cost. Comparing to traditional small molecular drugs, the peptide drugs has high target specificities as well as high target affinities. And when comparing to traditional therapeutic proteins and biologics, the peptide also have better membrane permeabilities, and also the peptide is less immunogenic. For these reasons, more and more focus has been derived into peptide drug discoveries. And that's the reason why WuXi are developing our own peptide drug discovery platform. So this is an overall timeline summary of how we develop our peptide drug discovery capabilities. Starting from 2020, we launched our first version of peptide drug discovery platform, which including the peptide DAO version 1. In this first version of peptide drug discoveries, we include one libraries, which has 4.5 billion diversities, and it covers 9 amino acid ring size. After 3 years of the experience accumulations, we launched our second version of peptide DNA-encoded libraries, which now including 5 different libraries, 200 billion chemical diversities, and we cover the ring size from 6 to 9 amino acids. In the meantime, we recognize that the DNA-encoded library could not fulfill all potential screening purpose. That's the reason why we also establish our phage display first versions, which also have 5 different libraries, more than 5 billion diversities, and the ring size is very diverse from 9 to 30 amino acids. After a year, in 2024, we developed our MI display platform, which including 8 different libraries, 8 trillion chemical diversities, and it covers the ring size from 10 to 17 amino acids. And this year, today, we are happy to launch our third version of peptide drug discovery platform, which including peptide DAO updates, phage display updates, and the MI display updates. In peptide DNA-encoded library, third version, now we have 12 different libraries, including diverse libraries and the focus libraries. Now we cover more than 300 billion diversities, and we are now even making the ring much larger. Now we can cover the ring size from 4 to 11. Also, for the phage display, the second versions, now we have more than 27 different unique structure libraries. We have more than 15 billion chemical diversities, and we covered ring size from 9 to the 13. For the MI display, the second versions, now we have more than 66 different library structures, and we cover more than 15 trillion chemical diversities, and we further expand our monocyclic peptide ring size from 10 to the 17 to 8 to 23. We believe these upgrades could further help our clients to get better and more successful hits from peptide drug discovery. So what's so unique about these 3 different technologies? So we believe DNA-encoded libraries and the display technologies are all very good technologies for de novo peptide discoveries, and they all have their unique strengths regarding different application scenarios. Let's begin with the peptide DNA-encoded libraries. So for the peptide DNA-encoded libraries, the library diversity is about 10 to the 11, and now we have around 10 more off-the-shelf libraries. One unique strength of the peptide DNA-encoded library is the delivery time. We can finish the screening and data analysis within 4 weeks. And also, in the peptide DAO, we can include more than 1,000 natural amino acids to further expand the diversity of this chemical space. For the MI display libraries, the most unique feature of it is that the library diversity is super big. As we can see on the figure, the library diversity can reach 10 to the 12, and now we have more than 50 different unique structure off-the-shelf libraries. The lead time of the MI display is relatively longer, around 10 weeks, and now we can provide more than 20 different cyclization strategies to meet our clients' request. For phage display, it also has its unique advantages. For example, we can do cell-based pannings, and we can even do in vivo pannings using phage displays. And now we have more than 25 different off-the-shelf libraries and the delivery time of the video display is relatively short. So let's begin with the MR display to basically introduce what we could do with the MR display and how we can really apply it in early peptide drug discovery. So on the left part, it shows the general workflow of MR display, starting from a double-strand DNA templates. We do in vitro transcriptions to transcribe the DNA into its corresponding RNA template. And then we do pyrimidine ligations to ligate a linker together with pyrimidines to the mRNA template. Then with this mixture, we do in vitro translations to translate corresponding amino acids and make it into a peptide. So one critical step of the mRNA display is the cyclizations. We can use traditional disulfide bound based cyclizations, but we can also adjust the cyclization methodologies using other small molecular linkers. After the cyclizations, now we have a library that is ready to use for the selections. Before we actually do the target protein selections, we're going to do several times of the negative counter selections using either native beads or counter proteins. This is trying to remove all non-specific binders that we don't want. After we finish the negative counter selections, we're going to moving forward into the actual target selections. By immobilize our target protein on the magnetic beads, we incubate our libraries with target protein and then wash away any non-binders. After that, we can recover what's left on the protein as our potential hits. We're going to repeat the whole cycles for 5 to 10 rounds. We can then identify which peptide binding to the target protein by using high throughput data analysis. So on the right part, this is showing a wonderful examples of how we use the mRNA displays to looking for allosteric binders, which not only have strong binding affinities, but also binding to an allosteric pocket. As we can see, these hits we identify, we call them peptide 8. When we measure the binding affinities using SPR assays, we can see it shows single-digit binding affinities to its target proteins. And when we further incubate the system with the natural binding ligand to oversaturate the target protein, this peptide 8 still binds to its target protein with 2-digit nanomolar binding affinities, indicates it was binding to some other pocket comparing to the natural ligand. So to summary, for our mRNA display platform, we have more than 1,000 billion chemical diversities that can be used per screen, and we have more than 50 different libraries with unique peptide structures, and we can even customize our library structures based on the actual project needs. And in every screening campaigns, we're going to analyze more than 10,000 different amino acid sequence for clustering and the enrichment analysis to help identify the most potential hits. And also, we have been developed a lot of strategies for pocket or target-specific binders identifications. So in the next part, I'm glad to share some of the updates regarding peptide DNA-encoded libraries. So from the last year in 2024, we already have more than 500 amino acids used in our peptide DNA-encoded library constructions, and we can do 5 different cyclization methods with 22 different linkers. And we have about 12 libraries ready to use, and the amino acid ring size is up to 8 amino acids. But we feel like this is not enough. That's why we keep expanding our capabilities and doing our internal R&Ds to see whether we can build more powerful peptide DNA-encoded libraries. So in this year, we have been used more than 1,400 amino acids into our peptide DNA-encoded libraries. And now we have 10 plus cyclization methodologies, and we have more than 30 different linkers that could be used to cyclize the peptide. So together, these two stuff and the improvements help us greatly expand the chemical diversities of the peptide DNA-encoded libraries. Due to these internal R&D improvements Now we have around 25 different libraries that is ready to use, and now we can construct a peptide DNA-encoded libraries up to 17 amino acid ring size, which is highly suitable for any potential purpose of the screening projects. So in these slides, we summarize what we want to show in this launch event regarding our new versions of the peptide DNA-encoded libraries. We would like to show you guys what we actually cover in these 300 billion diverse peptide libraries. So first of all, we have around 62 different billion diversities linear peptide libraries, and we including different lengths of the linear peptide to further increase the chemical space. Also, cyclic peptide libraries is also a hot topic in the market. That's the reason why now we have 13 different cyclic peptide libraries that is ready to use, and the total diversity is around 177 billion. In these cyclic peptide libraries, we have a huge diverse amino acid collections, and we also use a lot of different cyclization strategies. We also focus on some special design of the peptide DNA-encoded libraries, such as small cyclic peptide libraries. In these libraries, we try to make the library very small. The ring size is pretty small so that it is increase the possibilities of the oral absorptions and follow the guidelines of the R5 chemical space. For these small cyclic peptide libraries, we have two different library designs, which including about 52 billion total diversities. Another focus library design we are trying to develop and successfully launch is the RDC warhead cyclic peptide libraries. Based on the literature research and our previous project experience, we designed these RDC focus libraries based on different chemical properties, such as better water solubilities, better target affinities, and also fast excretions. We currently have 2 RDC-focused libraries, and the total diversity is around 10 billion. So the last story, but which is the most exciting story I want to share with you guys, is that we first time prove that the display technologies can be combined with the peptide DNA-encoded library technologies for hit-to-lead optimizations. So in these case studies, we have an initial priming hit funded by phage display technologies. And we want to further improve the affinities. That's why we're building up our own peptide encoded library focus libraries, which incorporate more than 700 different building blocks in each cycle, which definitely increase the chemical space that we scan in the screenings. And after we're building up these focus libraries, we do DNA encoded library screenings using these focus libraries, and we pick several of the highly enriched hits for further SPR validations. What we find is that in these validated hits, we can see some very potent hits. The best hits we can see, which actually increase the affinities for more than 300-fold comparing to the initial phage display hits. Overall, this story tells us that we could use peptide DNA-encoded library as downstream hit-to-lead optimization strategies followed by display technologies.
Thanks for the interesting stories on how we combine DAO and display technologies to empower the peptide drug discovery. Now we have learned a lot on our comprehensive and advancing hit finding platform. On the other hand, identifying a promising target is crucial for success in early drug discovery as well. Now let's welcome Zhongyao back to unveil our capabilities in aiding the discovery of an optimal target.
Thanks for the introduction, and it's my great pleasure to also present the integrated target discovery platform. Today, we're gonna talk about how we use the target discovery capabilities in not only target discovery, but also target deconvolution and target validation. So for the integrated target discovery platform, currently we have more than 1,800 animal model data sets, and so far for the previous all those years, in together we have been validated more than 100 different new targets. And we have in-house validated more than 50 in vitro and in vivo screening models, which is ready to use for future new target discovery. So what is target discovery and what's new about our target discovery platforms. So in our platform, we want to not only help our clients to identify new target, but we also want to provide one-stop solutions from target discovery all the way to target trackability assessment. For example, for target identifications, if there's already a ligand coming from phenotypic screenings, we can help to do target deconvolutions to find out the potential target within the cells of this particular ligand. So if we do not have a ligand and looking for a brand new target, then we could use either in vitro screenings and in vivo screenings to manipulate the phenotype of our models to looking for a novel new potential therapeutic target. But the screening is only the first step. We have to do a very comprehensive assays to validate this target in not only in vitro models, but also in vivo models. Another thing that we want to put emphasis on is the target trackabilities. A lot of target is highly relevant to a certain disease phenotype, but unfortunately, not a lot of ligand can bind to it, which make it super hard to make a drug. So we also develop our in-house DNA-encoded library screening database, which we call Vision, to help our client to assess the target trackabilities. For our target discoveries, on the left side, showing our selected successful stories of all those years, we have been working closely with our client, and we proudly publish these new target finding stories with our client in these publications. So far, we have rich experience, including a lot of cell lines in different cancer subtypes, and we have a lot of ready-to-go Cas9 stable lines, which means we can start the screenings very quickly. Now also, as we described before, we have a lot of different well-established screening models, both in vitro and in vivo, so that we can directly using these screening models combined with different selection pressures to looking for a certain new target. And also, we have developed a very good SOP-guided cell line characterization workflows to make sure these targets identified can be repeated, reproduced, and validated. Besides target discovery, target deconvolution is also a very important part. In WuXi biology, we try to combine the assay with proteomics capabilities to help do compound or let's say ligand deconvolute. So let's say we have a compound, then we can, using our special mechanism capabilities, to do some optimization and designs to make it into a modified compound, which has a photo cross-linking moieties. And then we're going to incubate this compound with the cell lysate and do photo cross-linkings, and as well as biotilations. After all those processes, we're going to do pull-down assays and send the mixture into our quantitative proteomics analysis. By using our strong proteomics capabilities, we can deconvolute which protein actually binds to this ligand, and then we can even understand whether there's any potential off-target risk. And on the right side is these examples which we use a tool compound to identify not only its potential targets, but also its potential off-target. So overall, this is a one-stop solutions for ligand design, cell assay, and quantitative proteomics. As we described on the beginnings, finding a good target is only the beginning of the drug discoveries. Sometimes we can identify several good candidate genes, which all shows a highly relationship to its target disease phenotype. How we can make prioritize these targets based on the target itself. This is actually one of the amazing application scenarios of the DNA-encoded libraries. By directly using the billion-level chemical space of DNA-encoded library and the high-throughput screening approaches, we have been generated more than 3,000 protein with ligand interaction database. Using this database, we can not only evaluate potential chemical structure binding selectivities, but also we can evaluate this target druggabilities by looking into the database and to see what kind of compound actually binding to these proteins.
Thank you to all the speakers for the fantastic talk. During the event, we have received many questions. I already picked some representative ones. Maybe let's start from the target discovery. Zhongyao, there's a question for you. So for target deconvolution, we combine the proteomics together with fully cross-linking with potential target. So is there any application scenarios for the target deconvolution?
Thanks for the questions. And this is actually a very good question. For target deconvolutions, we have a lot of different toolkits which is suitable for different kind of scenarios. In the webinar, we show how we could using photo cross-linking based methodologies plus proteomics to deconvolute the potential target. And in the other hand, we can using other methodologies such as proximity labelings or phenotype manipulation based cellular assays to deconvolute the potential target of a good ligand.
Thank you, Jungyao. Then I will invite Wenji to address those DAO and small molecule discovery questions. The first one, it seems that your screen products, the Dell Pro and Dell Lite, have been updated with many new libraries this time. Could you please explain the rationale behind incorporating these new libraries into your screening products?
As mentioned, the new libraries we added are 3-cycle libraries for better drug likeness. To achieve this, we typically curate hundreds of building blocks for each reaction cycle, resulting in a library with a total size of around hundreds of millions. Meanwhile, we aim to ensure that libraries within the same product are not too similar. Therefore, when designing libraries for different products, we use cheminformatics method to check for diversity. For Dell Pro, we incorporate WuXi proprietary building blocks and chemical reactions, while for Dell Lite, which we aim to use more commercially accessible building blocks, this makes it easier for our customer to follow up by themselves. Ultimately, our goal is to balance drug likeness and effective coverage of the broad chemical space while maximizing screening efficiency for each product.
Okay, thank you, Wendy. So there is another question also about the libraries. What upgrades have you made to your HTS libraries compared to the previous version?
So we have added over 100K new compounds with high diversity ensured by cluster analysis. It contains self-designed and synthesized compounds, proprietary scaffolds, and molecules. We apply strict metachem filters, including PINs, RIOs, and cassias, which is more stringent than most of the commercial screening sets available in the market. And besides that, we also include 69K covalent molecules, which targets multiple amino acids beyond cysteines.
Thank you for answering those two library questions. There are two fancy updates attract a lot attention today. So one is for the Dell Zipper. So audience want to know about, is the Dell Zipper only applicable to Dell libraries produced by WuXi?
No, it should be able to be applied to most Dell libraries available in the market. Dell Zipper is our new screening methodology, and it requires multiple processing steps, both before and after the screening. The application of Dell Zipper doesn't really depend on the original design of the library, so we believe this is actually a very universal solution to all libraries available.
Thank you. The other is for the D2B. So regarding the miniaturization of D2B, which assays have you tested it on and how effective has it been?
We have tested this method on various different assays, including HTRF, HIBIT, CTG, ASMS, and HCS. We also tried this method on several druggability profiling assays, including toxicity, permeability, and stability. And we have also developed method to test the impact of impurities and byproducts on the assay readouts. The results from these assays are orthogonally validated, showing high consistencies in the activity ranking of the compounds. So we believe in the robustness of the assay format.
Thanks, Wenji, for addressing those questions. Jungyao, you shared several methods on peptide discovery as well. There is a question that regarding these three different screening methods, in which screening technologies you guys have the most successful rate, or in a brand new target, which screening method should we choose?
Okay, thanks for the question, and I would say this is a very, very good question. And actually, we got these kind of questions from our client a lot. So, like I said in the webinars, the peptide DNA-encoded library, the MR display, and the phage display all have their unique advantages. And actually, in our previous project experience, we do observe that in some target, we see better selection performance in peptide DNA-encoded library, while in other potential targets, we see a better selection performance in MR display. So my first answer would be if it's a very challenging and brand new target, you probably want to try both peptide DNA-encoded library and MR display. And the second thing is that you could really choose the screening methodologies based on the ring size and the amino acid compositions. So if you would like to larger rings, nature amino acids, MR display is definitely the way you want to go. And if you're looking for smaller ring size, unnatural amino acids, and with a smaller ring but have a better affinity, then probably peptide DAO is the one you want to use. Thank you.
Thank you, Zhongyao. So we have time for about 2 more questions. Andy, discovery paths of oligo may be different from the traditional ones. So audience wants to learn about how long do you need to come up with an oligo preclinical candidate?
Yeah, thanks a lot for that question. So the typical timeline from oligo sequence design all the way through preclinical candidate is about 1 year. This includes all the in vitro screening as well as in vivo testing in rodents and a simple NHP model. Of course, this is really dependent as well on the specific target and disease. For some disease models or targets, multiple rounds of optimization of the compound may be needed, and so that might lead to additional time, right? For example, we can optimize the molecule for increased potency as well as increased safety, right? Meaning reduce off targets. We can also try to optimize the delivery, right, if needed.
And do you have any specific suggestions for developing oligo drugs for CNS targets?
Yeah, that's another great question. So our team has accumulated a lot of experience in different disease areas, so we can offer, tailored suggestions for different, disease areas. So coming back to CNS, I'll, discuss this in terms of the 3 points that I raised in my talk, which is mainly efficacy, delivery, and toxicity. So in terms of efficacy, we can use appropriate cell models for the CNS so that we can screen for the best compounds, right, that would have efficacy against CNS. So this includes like iPSC-derived neurons as well as iPSC-derived astrocytes, microglia, etc. In terms of delivery, right now the state of the art is to deliver the compounds through intrathecal injection. In the case of siRNAs, you might need to couple the siRNA to a delivery ligand, such as a lipid, to help it enter the cells. But nowadays, there are additional efforts to deliver oligo drugs across the blood-brain barrier into the CNS. So this will be more convenient for patients. So we can support this by helping to develop like antibody oligoconjugates or peptide oligoconjugates that can cross the blood-brain barrier. And then the third aspect is toxicity. So for CNS toxicity, we offer various types of assays, such as in vitro calcium oscillation assays to assess for neurotoxicity. We can also do the in vivo neurotoxicity assessment, namely using the functional observation battery, right, or FOB for short.
Thank you, Andy. Thank you, Zhongyao and Wendy as well. We can follow up more questions offline, so we appreciate your questions and insightful answers from our speakers. We hope you are as excited about our new offerings as we are. Stay tuned for new updates and innovations. Have a great day and see you next time.